All right, guys. So this is regarding the Lac Operon, and before we get into it, I would like to discuss where exactly we are at in the cell. So many may know the rRNA is just a fancy word for a ribosome, right? The bigger piece here and the smaller piece here. And then we know of mRNA or messenger RNA. Usually it's sandwiched between the ribosome. Ultimately, the ribosome, um, together with the mRNA and um, let's say and other helpers, they may ultimately make enzymes and proteins. So for now, these are the operon or mRNA, but just to get an idea of where we're at, the purpose of why we need to discuss this scenario of, let's say, the Lac Operon is, let's say that, well, microbes do not prefer to metabolize lactose, which is a big sugar. They naturally would like to metabolize simpler sugars like glucose. So if they had a choice, they would always go for glucose. But what if there's only lactose in the environment? Then if there's only lactose in the environment, then the cell, like for example E. coli, will need to figure out a way to, hey, turn on a system so they can metabolize it, use that resource that they have available. And then they also need to figure out a way to turn off that system, because they can't have that lactose breakdown continuously if there's no lactose in the environment. It's just a waste of resources. So we need a way to turn it off. And that we'll discuss this in 3 simple scenarios. What happens when there's no glucose in the environment? What happens when there is lactose? And then how can we amplify this effect? Lactose metabolism, or scenario 2. And then what happens when there's no lactose, which is scenario 3. So let me break down these pictures. So now scenario 1, no glucose, but there is lactose. The inactive CAP, I have a typo here, I apologize, but it's catabolite activator protein binds with CAMP. And the concentrations of CAMP, it goes up because it acts as an alarm that glucose levels are low. So inactive CAMP binds with CAMP, which makes this protein active. So the active CAMP guides RNA polymerase into the promoter region of the Lac Operon. So this is the Lac Operon here. It may help that RNA polymerase has P in it. So think of P for P on the promoter region. So RNA polymerase only binds to the promoter region of the operon. So when it binds to this area, our RNA polymerase can now transcribe 3, for now, let's say 3 specific genes, the Yas genes. I just remember that as let's say my cousin's name, Yasmin. The Y gene is a recipe to ultimately make permease. So it doesn't make it right off the bat. It needs certain steps. But again, genes are just a recipe. I think of also the permease. Its function is to transfer lactose into the cell. So I think of the Y gene as a slingshot. So the Y gene ultimately makes, it's a recipe to make permease, and it slingshots lactose into the cell. The A gene is a recipe to make transacetylase, which metabolizes disaccharides. The Z genes is a recipe to make beta-galactosidase, which breaks down lactose into simpler sugars, glucose and galactose. So that's scenario 1, no glucose, but there is lactose. So now we need a way to amplify that effect. This is scenario 2, lactose metabolism. Lactose from the extracellular fluid or outside the cell gets slingshot into the ICF or intracellular fluid or inside the cell via permease. And then lactose gets converted into allolactose. Allolactose binds to the repressor proteins. So you guys may see this formula that I have here going on. It's just a plus sign. That just means that whichever first word is gone here, they're gonna bind to each other. So allolactose is gonna bind to the repressor proteins, which will make the inactive repressor proteins. Essentially, these guys turn inactive. So they float away from the operator region. The RNA polymerase can continue to sit on the promoter region, so it can continue to transcribe the Yaz genes, the recipes to make certain enzymes that we just went over. Basically, essentially, amplifying scenario 1, right? It just continues. Scenario 3 is no lactose. So we need a way to turn off the system to save resource. And the way to do that is that tryptophan or TRP concentrations go up, and it is very important to know that it is produced by the structural genes, specifically A, B, C, D, E genes. The tryptophan binds with the inactive repressor proteins. Just know that the inactive repressor proteins are made via the I genes. So tryptophan binds with the inactive repressor proteins, which makes this protein active. Active repressor protein then now binds on the operator region. The RNA polymerase now gets booted off the promoter region, so it stops lactose metabolism in order to save resources. So I think of this as a button system, and then it'll make more sense in a second when I go back to the larger picture. Just know this is the TRPR prawn. Let me go back to that picture here for you guys. Apologize there. There we go. Let me review this, and then this may make a little bit more sense, hopefully. All right. So in a scenario where there's no glucose, the inactive CAMP or catabolite activated protein binds with CAMP. The concentrations of CAMP go up because it acts as an alarm that glucose levels are low. The inactive CAMP binds with CAMP, making this protein here active. Active CAMP guides RNA polymerase onto the promoter region. So RNA polymerase can transcribe the 3 genes. The Y gene is a recipe to make permease. Permease it's an enzyme, hence the ASE at the end. It's an enzyme that functions as by transferring lactose into the cell. The A gene is an enzyme. The A gene is a recipe to ultimately make transacetylase, which metabolizes disaccharides. The Z gene has the recipe, or is the recipe, to make beta-galactosidase, which breaks down lactose into glucose and galactose. So just know that genes, they are not enzymes. They just hold the recipe in order to make proteins. Which I almost did that mistake there. So now scenario 2 is just amplifying this effect in a way, I guess. It's lactose metabolism. Lactose gets slingshots into the cell via permease. So allolactose binds. I guess going back one step, lactose gets converted into allolactose, and allolactose binds to the repressor proteins. Then it makes this protein here inactive. So now the inactive repressor protein floats away from the operator region. And this picture here, it was something I drew just to explain something else. So just ignore this picture. I apologize. But yes, the inactive repressor protein just floats away from the operator region, and it floats away. The RNA polymerase can continue to sit on the promoter region, so it can continue to transcribe the Yaz genes. Just know all genes are transcribed and translated ultimately to make proteins and enzymes. It's a little safe haven here. The scenario 3 will be no lactose. So we need a way to turn off the system in order to save resources again. So TRP or tryptophan, the concentrations of it go up, and it is produced by the structural genes, specifically A, B, C, D, and E genes. The inactive repressor proteins are produced via the I genes. That's very important to know, or the recipes, how these guys are ultimately made. So the tryptophan binds with the inactive repressor proteins, which makes these guys here active. The active repressor protein binds to the operator region. So again, when I mentioned it's a button system, the way I think of it is because since the active repressor protein sits on the operator region, so let's say boom, so it pushes this button down, which pops this button out, right? And then out, which boots RNA polymerase off the promoter region, so it can stop the lactose metabolism scenario in order to save resources. And that is the end of the Lac Operon and this TRP Operon introduction. If you have any questions or if there's any discrepancies, please send me a message. Thank you.